In interphase, a nuclear envelope bounds the nucleus, which contains one or more nucleoli. Two centrosomes form by replication from a single centrosome, which features two centrioles. The chromosomes duplicate during S phase, but are not yet condensed. In prophase, the chromatin fibers become more tightly coiled, condensing into discrete chromosomes. Also, the nucleoli disappear, and each duplicated chromosome appears as two identical sister chromatids joined together. The mitotic spindle begins to form, and the centrosomes move away from each other, and a microtubule forms between them. In prometaphase, the nuclear envelope fragments. The spindles now interact with the chromosomes, and the microtubules extend from each centrosome toward the middle of the cell. Each chromosome now has a kinetochore, and some of the microtubules attach to the kinetochores. In metaphase, the chromosomes are at the opposite ends of the cell, and the chromosomes lay on the metaphase plate. The kinetochores of the chromatids attach to the kinetochore microtubules. In anaphase, the sister chromatids separate and become chromosomes. The chromosomes move towards the ends of the cell, and the microtubules shorten. Also, the cell elongates. In telophase, the nuclei begin to form, and the nuclear envelope begins to form from parts of the original parent cell's nuclear envelope. Also, the chromosomes become less dense. In cytokinesis, the cytoplasm divides. Also, the cleavage furrow forms, and the cell divides into two.
The main difference between mitosis and meiosis is that meiosis is basically mitosis, but it's happening twice to form 4 daughter cells with only a haploid number of chromosomes. Being shown now is interphase 1, and it precedes meiosis, and it's where the chromosomes duplicate. Prophase 1 is longer and more complex than in mitosis, and occupies 90% of the time in meiosis. Chromosomes begin to condense, and the homologous chromosomes come together to form tetras of 4 chromatids. The initial random arrangement of these chromosomes helps increase the amount of variety in the genetics of the daughter cells. These chromatids are crisscrossed to help hold them together, and this crossing over portion provides a lot of genetic variety. Meanwhile, the rest of the cell prepares for the division. Metaphase 1. The chromosomes are arranged in the metaphase plate and still in the pairs, and the kinetochore microtubule is attached to one chromosome of each pair. Anaphase 1. The spindle moves the chromosomes toward the poles, however, the sister chromatids remain attached and move as a single unit. Telophase 1 in cytokinesis. The chromosomes have moved to the poles. In some species, they recondense and the nuclei reform, and there is an interphase 2. In other species, they go straight into preparation for the other division. There is no replication of the DNA at this time in all species, however. Prophase 2. The spindle forms and the chromosomes move to the metaphase plate. Metaphase 2. The chromosomes are positioned on the metaphase plate again, and the kinetochores of the sister chromatids face opposite poles. Anaphase 2. The sister chromatids separate and they move toward the opposite poles. Telophase 2 in cytokinesis. Nuclei reform and the cell splits again with the haploid number of chromosomes.